Thanks, Nick. So as Nick mentioned, we have been on break. So just wanted to give, if we have any new people joining us, this has been a really enjoyable Coregonine science webinar that I think, based on the feedback we've gotten, we now have done, I guess Trevor will be our 14th webinar. Started April of 2021. We've had really good attendance. Most of them, the speakers have given permission for us to record them and to archive them on the GLFC YouTube website. Maybe Nick could even drop that in the chat if you wanted to check that out. The diversity of topics, ecology, evolution, reintroduction challenges, with a hatchery sort of perspective, monitoring talks. Of course, a lot of them are Great Lakes based like we'll have today, but we've had a lot of sort of international Coregonine flavor as well. Alaska, Finland, France, Ireland. And so we're looking for speakers. This is a call. We're looking for volunteers to speak starting in December and onward. We are looking forward to next month's seminar. Dr. Feline Fuellner, I'm probably not getting that pronunciation right. She's going to be talking about reverse speciation of whitefishes in Lake Constance, which we think has really nice sort of implications and some similar questions we're thinking about in Coregonines. And that's actually a really nice segue to introducing today's speaker, Trevor Krabbenhoft. Let me stop sharing now. So Trevor, I think many of you in the Great Lakes may know of him. Trevor actually got his bachelor's degree in zoology at North Dakota State University, and then went down to the University of South Carolina for his master's for a biology degree, and then he's really traveled a lot. I forget how many states he told me, 10 states now. Is that right? PhD, University of New Mexico with Tom Turner. And Trevor just shared with me that is where he got turned on to this idea of ecology in addition to evolution. And I think that's one of the things I really value about Trevor, is that certainly he has great genomic and evolutionary skill sets, but he always puts that in a really nice ecological perspective. And when you sort of read about his website, I think this is a nice summary of what Trevor does. He talks about using evolutionary and ecological approaches to understand how fishes respond to environmental change in any kind of aspect: climate change, invasive species, contaminants or nutrients, overfishing. And he seeks to address these questions not only at local scales, like where he is now in the Great Lakes, but also globally as well. So we're really pleased to have Trevor here today. And if you want to pull your slides up, Trevor, his presentation will be about 40 minutes or so. And then, as with previous speakers, Trevor's willing to stay on and hopefully have a really informal question and answer session following. So thanks very much, Trevor. And we'll chat in just a little bit.
Thank you, Bo. I appreciate that thoughtful introduction. And thanks to Bo and Nick and others for setting this webinar up. This has been a really fun series. I've learned a lot in the process. So I'm really looking forward to next month as well. So it's going to be fun. And thank you all for coming, taking time, especially at the start of a semester or maybe tail end of a field season, to get organized a little bit. So my talk today, as Bill mentioned, is on evolution of the Laurentian Great Lakes ciscoes. I'll try to, I say Laurentian because I know there's some folks from outside of the basin joining us, so trying to be specific about that. And I'll talk about some of the evolution of these fishes and try to put that in a little bit of context for fisheries management and restoration. So today's group, most of you are intimately familiar with these fishes, but I know we have some people from other systems here today. So we're talking about ciscoes, genus Coregonus. Some people call this the subgenus Leucichthys. This is a little bit of the diversity that's extant. These are wonderful illustrations from Joe Tomalleri. And I have this photo of Nathan Backenstoss as a reminder to to mention that much of the data I'm presenting is part of Nate's PhD dissertation. I presented this back at some of this research at the Joint Aquatic Sciences meeting, and I left Nate off the title slide inadvertently. I'm sorry, Nate. So here's Nate's proper acknowledgement. Nate's also on the postdoc market after he finishes his PhD. So I'll start there. I'll give a very brief introduction to ciscoes. Most of you know this story already, but this group of fishes once supported a really vast fishery in the Great Lakes. Of course, this wasn't sustainable at the levels that you see here. So that was the first sort of threat to ciscoes. The other one was invasive species. So invasive sea lamprey, for example, played a role in the decline of Great Lakes ciscoes. And of course, this is a bad place to park your cars. Back in the day, the industrial scale pollution and reduction in water quality also probably contributed to some decline in sea lamprey. But to some extent, all of these factors are being taken care of much better than they were historically. So better fishing regulations, better pollution control, Clean Water Act was really useful, and programs to reduce invasive sea lamprey that are ongoing and important control. So it's a good time to think about restoration. But we've lost some of the diversity, the original diversity. For example, Lake Michigan. You've seen this slide from Celts before, many of you. Many of the deep water species forms were either extirpated or are now extinct altogether. But there is some remnant diversity, and the challenge now is how to take that diversity and see what's possible in terms of restoration and conservation. But the scale of this is enormous. I always like these space photos of the Great Lakes showing the scale. Not just physically, but also across different jurisdictional boundaries and lots of people involved in this process. So it's very complex. Fortunately, there's a good plan in place. This is the science-based approach to restoring Krabbenhoft. This restoration framework has been put into place, and it's been a really useful tool for trying to organize and make plans, science-based approaches toward restoration of Great Lakes Krabbenhoft. So this involves science, collecting data, developing restoration plans, and implementing those plans. And it's an adaptive approach. So as more data, more information comes in, we can people can modify the plan and how we respond to that new information. So people in the Great Lakes cisco world know this plan really well. But for those of you outside working on other systems, I think this sort of framework could really be adapted to other systems as well. So I'd encourage you to check it out. Much of what I'll talk about today is focused on this top right box, this effort to resolve Krabbenhoft taxonomy using genetics and ecology. And a little bit more broadly is to understand some of the diversity that's still extant, as well as some of the historical diversity that's present, and use that as to contribute to this sort of restoration framework. Some of the questions I'd like to focus on, what are the patterns of variation, genetic variation, morphology, and ecology? So I'll present some data there. Ask this question of concordance. It's much easier, it makes our life much easier if these different data sets are in agreement. So hopefully we can find concordance across data sets. I'm talking today a lot about evolution and thinking sort of in that evolutionary context. What are the drivers of diversification? And I think this is an important question to understand what caused this diversity that we had, at least at one point. What were the processes that gave rise to that diversity? And that's useful because we can then understand if we still have those same processes in place, or if we need to do something differently with respect to restoration. And also the timeline to understand where we're going now and into the future, having a historical perspective. How long have these different forms been present, and what were the conditions that gave rise to them might be useful. So I've shown this slide several times. I'm feeling a little sheepish about it, that I'm an early career researcher, but one of the hallmarks of becoming a mid-career researcher is that you show the same slides over and over again. So looking at this optimistically, maybe this is inching toward that goal, aspirationally maybe. So this is some of the diversity that's extant in, for example, in Lake Superior, where forms or species fared better from some of these events. 4 of the species I'll talk about today, Coregonus ardidae, hoyi, kaii, and Xenithicus, are shown here. Some interesting phenotypic diversity. It's probably related to ecological function to some extent. So differences in eye size and gill rakers, for example. The first thing we set out to do is try to understand some of the how this phenotypic diversity relates to patterns of genetic and ecological diversity. Excuse me. So the first place we started was with transcriptome data. This was led by Moises Bernal, who was a postdoc working with us. Now on to greener pastures, an assistant professor. We collected transcriptome data, and we specifically selected transcriptomes because we wanted to. We didn't have this. We couldn't do the whole scale of genomes, so we started with parts of the genome that were involved in the ecology. So we sequenced transcriptomes, and we're looking at patterns here. This is a PCA of transcriptome variation. I've shown this a number of times as well. So the letters, each individual letter corresponds to an individual, and the A's correspond to ardidae, kaii, and then hoyi and Xenithicus. And so it's pretty strong differentiation, except for these 2, hoyi and Xenithicus. So that's good. We have some genetic differentiation. And morphology actually tells a pretty similar story. So species that are distinct in terms of transcriptomes are also distinct in terms of morphology. A little bit more noise here. There's phenotypic plasticity also in morphology. So there's maybe some differentiation there, or cause for more overlap. And then we did bulk stable isotopes, carbon, delta-13 carbon, and nitrogen. And there's better approaches, of course, now than there were at the time, but this is what we did. So we see in general, there is some concordance across these different data sets. Individuals that are distinct genetically tend to be distinct morphologically as well. All right. Because they're transcriptomes, we can look at what those genes actually are. So so we did that at the time. We didn't have a genome assembly, so we just lined the SNPs up. These are single base pair changes, is what I'm referring to here in SNPs. These are base pair changes that differ across these nominal species of ciscoes. Okay, so most of the ciscoes do not differ. Most of the SNPs do not differ in these cisco genomes. There's very little differentiation. But on the y-axis here, I'm showing FST, our measure of how different these how consistent these genetic variants are with taxonomy. So the higher up on this y-axis means there's more genetic differentiation. Some people don't like this term taxonomically informative, but I only just mean that these are genetic variants that are more strongly associated with a particular species on the y-axis. All right. So the genes that are of interest to us are the ones that are strongly differentiated, that tell us about the differences between species. So we can focus our attention here and look at what these genes are. We can look at these as maybe candidates for adaptation in this group. These are not just random genes. They seem to be associated with some of the phenotypic differentiation that we see. That's sort of a just so story. So we wanted to go in and actually look at some of these genes in more detail and try to validate that these patterns that we're seeing in a small data set are perhaps real. Katie Eaton was an undergraduate, did an undergraduate honors thesis with us, a rather ambitious one where she took some of these samples, much larger sample size, and used this nanopore sequencing approach to try to characterize some of these genes in more detail. So you can use PCR and make more copies of particular genes. In this case, she was focused on, we started with the gene that's most strongly differentiated. That was this gene called rhodopsin. We thought that made sense. It looks like it's under natural selection in terms of the transcriptome data, but we wanted to validate that. And while we were doing so, we decided to look at some of the other vision genes. These are involved in visual signal transduction. So these are some of our other candidate genes. This is read length. This is basically how long the gene is in terms of base pairs in our sequence data. So these peaks correspond to our sequence data. So you can see different peaks correspond to different genes. And then we can look at the patterns of genetic variation across ciscoes. So that's this paper from that Katie wrote. Just keep a long story short. Basically, the genetic variant that was really strongly differentiated in rhodopsin, this gene of interest, is shown here. This is a three-dimensional representation of what this rhodopsin protein looks like. It has these transmembrane domains, so the actual crossing the membrane, the cell membrane. And I'm showing here the black corresponds to DNA-based substitutions that differ across cisco species. So this one in the central part of this pocket, basically light comes into this protein and it causes a conformational change in the protein. So this strongly differentiated part of this gene is right here in the central part. So we think it's an important part of this protein. And we've looked at patterns of variation in this. So what I'm showing here for these species, I forgot to add, these are from Lake Superior specifically. Should have mentioned where my samples came from. Basically, these pie charts show the allele frequency in KD samples across species. So the chi is fixed for one allele, and the other three species are more or less nearly fixed for the opposite allele. So this is a differentiation. Also show here this lambda max. I mentioned this is a visual transduction molecule. So basically, in the eye, this is expressed in the eye and the retina, and as light comes in, this protein responds to particular wavelengths of light. And those wavelengths differ based on the actual sequence of the protein. People have done this experimentally, shine light of a particular wavelength into a fish's eye and see how it responds. So these numbers correspond to the maximal sensitivity of these particular proteins. All right, so chi differs obviously from the others. Here's the capture depth where these fish came from. The other three were relatively more shallow water samples for the most part. And then we have these deep water chi. So chi is a deep water species. It differs in this visual signal sensitivity in this SNP. So we want to connect that to light availability in Lake Superior. So this is actual wavelength data modeled as maximum wavelength that can be perceived by these fish. So if you go in shallow water, nearly the entire wavelength is still available to the fish in shallow water. That makes sense. Shallow water, there's not much light filtering, but as you go deeper in the water column, some of these red pigments or red wavelengths get filtered out as light gets attenuated as you go down into deeper water. So this blue appearance is because of this light filtering in deep waters. So chi being this sort of blue shifted allele is taking advantage of the light that's made available to it at these depths. And in contrast, these relatively red shifted, more shallow water forms are taking advantage of this extra light that's available to them. I think this is an example of adaptation to depth. So chi is the derived form, and this mutation may have allowed it to sort of take advantage of some of these deeper water conditions. Katie also followed this up by trying to provide some broader context. Is this SNP just by chance, or is this genetic variant here just by chance, or is there some kind of pattern across fishes? Is there something predictable about this? And so she took on this monumental task of taking the fish tree of life, shown here, 4,000 of the species at least, shown here. This is a phylogeny. And she took rhodopsin sequences from all these different fish species and tried to reconstruct the evolutionary history of those alleles. So all of these species have rhodopsin, and you can look at what rhodopsin looks like today in those species and then reconstruct the ancestral history of what likely their ancestors had. And then the lines then in this phylogeny correspond to what we think the ancestral state was, or the current state. And what you can see is that across the history of life, there have been many transitions between this red shifted versus blue shifted vision pattern across different taxa. So it's really interesting. Fish seem to be toggling back and forth between this red shifted or blue shifted light. This is identical amino acid substitution in these genes across very different evolutionary lineages, 300 million years of time. You see a kind of a hotspot. There's a lot of changes here, and this corresponds to the salmonids. So salmonids being anadromous, going from marine to freshwater. There's many transitions in the light environment, and they seem to respond accordingly. So it's interesting that evolution is normally an unpredictable process, but here's an example where you have really strong predictability at this particular wavelength. There's sort of constraint in how fish can see. And if you sort of stare at this figure for a while, it kind of starts to look like a fisheye. So if you worked hard on that transition. Other genes we've looked at, I'll just give a couple more examples. Another one we looked at that differs strongly across species is this gene called glutamate decarboxylase 2. This is, or GAD2. This is an obesity gene in humans, so it's often associated with obesity in humans and diabetes, and it's associated with lipid metabolism. Chi differs in this gene from the other species. And we thought that was interesting because of what we know about lipids and chi related in particular to buoyancy. So this is a paper from Krause et al., where they showed that chi is more specialized than hoi for diel vertical migration in deep water. So these lipids help with buoyancy. So we have these different levels of buoyancy across species. Chi, this is hoi, and artidae. So this might be another genetic variant that is directly associated with adaptation to deeper waters in chi. Okay, and this is found all the way in humans. Some of these genes do essentially the same thing across deep evolutionary lineages. So in humans, this is the diabetes sort of response pathway. Here's GAD. It's an autoantigen. It triggers this autoimmune response that causes diabetes. So it's really fascinating. You see these deep conservation of some of these genes. Okay, so those are a couple of the candidate genes that we've looked at. This approach, though, of just looking at transcriptomes and a relatively small sample size was sort of unsatisfactory to us. So we wanted to look more broadly across some Krabbenhoft diversity. So the next step, as part of Nate's PhD dissertation, we started working with Amanda Akis and others on whole genome resequencing of a much larger sample size, so a couple hundred individuals. And those data are shown here. There's a lot going on. We have samples mostly from Nipigon and Superior, a few Huron Lake fish present as well, and across several different species. So these are color coordinated as well. There's a lot going on, but what I'll draw your attention to is this boundary between Lake Superior and Lake Nipigon in this plot. So most of the diversity or differentiation is on PC1, and that's separates these two for the most part. So strong differentiation between these two different, across this boundary, probably corresponding to different glacial sort of refugia. I'll add, we had a few Huron samples, and just for those who are curious, these are Huron artidae that group with Lake Superior artidae. So I'll walk you through some of the species next in a little bit more detail. I'll break out Lake Superior and Nipigon. So for Lake Superior, if we zoom in on that part of the plot, we see a nice kind of triangular pattern, this data set. The one corner corresponds to Chi, these blue dots, nice tightly clustered. Hoi is the other vertex in this triangle. And then artidae is the last sort of part of this. I should say these identifications that I'm presenting here, these are not based on careful morphological analysis. This is just kind of sort of gestalt from both, field-based sampling. So some of these are potentially misidentified, but there is probably evidence for interaggressive hybridization for some of these intermediate individuals. The last species I'll mention is Coregonus denicticus, and you can see these, it's a little bit hard to see, but there's sort of a smattering throughout. And it may be that these are potentially misidentified and we didn't really sample Xenithicus, or it might be that there's some introgression happening and we don't have that species in its sort of pure form in this data set. It's difficult to differentiate between those given the data we have at hand. Just checking the time. And then if we look at Lake Nipigon, it's actually a slightly different pattern. So we have Artidae as, again, this triangular sort of pattern. Artidae is one of the species, but here we have an abundance of samples that are at least tentatively being called nigripinous in this case. So these are blackfin ciscoes. And then the third corner of this triangle is actually, in this case, seems to be mostly things that were identified a priori as Xenithicus. So these might be the real short jaw as one possibility. There's a lot more noise in these in terms of the identifications in this case. It's maybe more challenging. But nonetheless, we see strong differentiation across the different nominal species. All right, so that's genome-wide patterns of variation. And I should add that others have much more extensive data sets. So Amanda Atkis and others have much larger sample size and better geographic sampling. So, so there's more to say about that kind of variation. We've also started, we needed a reference genome to try to put these genetic variants in more of a context, a better context. So another part of Nate's dissertation work is sequencing and assembling the Coregonus artidae genome assembly. We've made some progress on that. These are some of the chromosomes in that assembly. It's an ongoing process, but I mention this now because we're at a point where it's, I think, useful enough to be shared with the broader community. So if anybody here is interested in using this, if it's helpful to your research, please reach out and we're happy to share this as a work in progress, but nonetheless get it out into the community. And I know several people are currently using it. We'll get it in NCBI, but we're still trying to optimize. There's a few issues with it still, but we've made good progress there. Some people asked if we could also just use the whitefish genome. This is lake whitefish, Coregonus clupeiformis. And part of the motivation for this is that these species, even though they're only about 9 million years divergent from one another, the genomes have been shuffled in a surprising amount. I was surprised when I saw this, but we compare the Artidae genome assembly to lake whitefish genome assembly. And this is basically each one of these boxes corresponds to a chromosome. So this is chromosome 1, 2, 3, and so on in Artidae versus chromosomes 1, 2, 3, and so on in clupeiformis. The dots on this plot represent individual genes, and these are homologous or orthologous genes across these assemblies. So if these genomes were perfectly collinear, you'd have a nice pairs of homologous chromosomes would be, these dots would fall perfectly on the one-to-one axis. You can see within a particular chromosome, there tends to be a lot of these sort of chromosomal rearrangements between. The genomes are surprisingly different. If you're worried about hybridization between lake whitefish and say cisco, I think given how different these genomes are, it might not be that much of a concern that the hybrids go on to back cross and so on. I could be wrong about that, but the genomes are very different, and so that makes it presents a barrier to introgression, genetic introgression between those species. Just as an aside. Now that we have a genome assembly, we can start to look at the actual spatial context of how this variation, genetic variation, plays out in the landscape of the chromosomes. So here we're showing the Coregonus artidae Genome assembly, these numbers correspond to individual chromosomes in the Coregonus artidae assembly. And then the y-axis, again, is our measure of genetic differentiation. These are for the whole genome data set that I showed a moment ago. So the higher up a genetic variant is on this y-axis, the more taxonomically informative or strongly differentiated that variant is across populations. One I'd like to show, and this is sort of a moving window across chromosomes. One I'd like to show is this particular region on chromosome 33. There seem to be these 4 data points that are sort of at the extreme end. So we want to know, in this particular comparison, this is artidae versus Kii. So we're comparing these 2 species. We zoom in on that region specifically. So we're now looking at just at chromosome 33. This is one end of the chromosome over to the other end. And I think it's actually cut off. Again, y-axis is FST. The dotted line here is sort of a moving window of differentiation. And I'll note, draw your attention to this peak, this highest peak in along this region. And this is FST at 0.75. These are very strongly differentiated regions. There's a lot of fixed differences or nearly fixed differences right in this region. It's about a 500 kilobase region, 500,000 base pair region, relatively small piece of a chromosome that's differentiated. And now that we have genomes, we can look at what's actually in this region. So we always like to see what those genes are. And you might not know these genes, but we're learning a lot about them, REPS2 and NHSA. These are 2 genes that are found in this region. They're strongly differentiated between Kii and artidae. And what do? They're involved in retinal development. So the actual development of the eye is the function. And humans have these as well. In humans, if there's X-linked vision problems, so in humans, these genes are on the X chromosome. And men, because they have only one X chromosome, tend to have more of these vision-related issues, including, in this case, these 2 particular genes cause Nance-Horan syndrome. This is congenital cataracts is the problem in humans. So it does the same thing, basically. In humans, it helps the eye develop, the retina develop properly. So that's pretty interesting that we see these strong genetic variants, again, corresponding to eye development. Also interesting is, I mentioned in humans, these are on the X chromosome. There's also a sex-associated gene. This is a male-associated gene in this region. So maybe some interesting links between sex and vision. I don't know much about that, but if anybody has studied this in ciscoes, I'd be really curious to know. So this is images from Eschenroder et al. This is Kii and this is artidae. If you look at these fish, what do you notice is really strikingly different between them? And its eye size is one of the first things that jumps out to me. So Kii develop this very large eye, which is energetically expensive in other fish species. People have done metabolic studies and have shown that the maintenance of a large eye is up to 15% of the energetic budget. Just maintaining this eye is energetically expensive. So it's costly. So you don't want to make a big eye if you don't have to. That comes at a cost to other things like reproduction and so on. So adaptation to depth may involve making a larger eye, collecting more light, for example, in deep water Kii. I don't have time to talk about it, but if anybody's curious, we've started looking at these similar kinds of what we think are chromosomal inversions in other taxa, other fish taxa, examples where fish are evolving across some light gradient. And surprisingly, actually, we found 3 other examples where these same genes, the same chromosomal region is involved. So we think this is maybe more of a fish-wide or vertebrate-wide pattern rather than just being unique to ciscoes. That helps us become more confident that there may be some adaptation actually happening rather than just chance events. I'm running short on time, but the last one I'll leave you with is another putative chromosomal inversion. This is a 12 megabase region of chromosome 3 we found. And this is 12 million base pair long, so a large chunk of a chromosome and it seems that bloater differs from the other taxa or Hoyi differs from the other taxa in this region. It's an example of, we think, a chromosomal inversion, so a piece of the chromosome that's essentially flipped relative to the other taxa. There's been there's some really fascinating older literature on this from Phillips et al. This is a paper from 1996, I believe, on chromosome structure. And they talk about some of this controversy about these karyotypic differences, these chromosome level differences. And they note this paper by Book et al. That found extensive chromosome variation due to inversions. These variations seem to confirm their status as distinct species. But Phillips, however, did not agree with this, so there's some controversy out there. But here's an example where we think we're finding some of these chromosomal inversions that may be important. So thinking to the future, this is an area I think would be really fun to look at, is to go back to some of these chromosome level comparisons and see what we can find with newer approaches. Okay, I'm running very short on time, so I'm going to skip the mathematics behind what I'm going to talk about. But basically, there's genetic tools now where we can look at how effective population size has changed over time. For example, if you have genetic bottlenecks over evolutionary time, we can look at we can model that with genetic approaches. I wish I could have gotten into that in more detail, but I can explain it if somebody's curious. So Nate and others have been working on this. So we took these genomes and we tried to we ran these analyses. This is coalescent theory. It's another term is historical demography. We can look at how effective population size, which is on the y-axis here, has changed over time. So from the current day going backwards 10,000, 100,000, or a million years. And what we found is that for a given, this is one individual of Artidae, we do some bootstrap replication. That's why there's variation. And you see that over the last million years, there was a population collapse up until a certain point, and then massive expansion. Try to speed this along. We did this same kind of analysis across 4 different species. The curves are remarkably similar. In large part because they share evolutionary history for at least part of that time. So I'll just put these all on the same sort of plane. These curves are completely overlapping if you go back a million or so years. But you get to a certain point and the curves start to diverge from one another. There's a low point. This low point corresponds from 19,000 to 33,000 years ago. Interestingly, that happens to correspond to the last glacial maximum. So the glaciers extended down, and not surprisingly, as these fish were in the refugial Mississippi and Atlantic population, refugia, populations were probably very low. The glaciers retreated. So this first light bar, 17,000 years ago. The Great Lakes are still covered in ice. Here's the maximal ice extent. But then the glaciers start to retreat. So by 10,000 years ago, the Great Lakes are now open for business. These habitats have been exposed, and now, surprisingly or not surprisingly, the populations expand in response to that new habitat. It's a really exciting result. If the habitat is there, then the populations can expand to take advantage of that. The divergence of these lines corresponds to essentially the point at which they become demographically independent. That's an interesting time period. They start to function as different units evolutionarily. And we think that took place about 65,000 years ago, just before the last glacial maximum. I'll very briefly do something I'm not supposed to do in the Cisco webinar series, and I'll talk about lake trout. This is the predominant predator, one of the native predators of ciscoes. Another adaptive radiation. And these are some wonderful illustrations from a paper by Andrew Meir and others, showing that there's similar divergence of different forms here in the Great Lakes and elsewhere. Ciscoes are not evolving in a vacuum. They're part of a food web. And so lake trout are an important predator. So these are the curves I showed you before for ciscoes. Using this PMC analysis, these are the cisco curves on the top. And we had, there was some data available for lean and ciscoette lake trout. So we took advantage of that and just ran it through the same analysis. And lo and behold, these curves seem to be coupled. Lake trout showed a similar population crash until the last glacial maximum. And then there's a slight lag. So as cisco populations expanded, lake trout populations also followed that expansion. That was really exciting to us because it shows that because cisco are common are an important forage species for lake trout, as cisco populations expanded, that provided, we think, a food source for larger lake trout populations. Going back to our wheel, much of the idea behind restoring carrageenan populations is based on this idea that if you restore carrageenans, that'll provide an important food web link, an important food source, for example, to some of these native predators. And the really exciting part of these data is that these data provide a historical precedent for exactly that. It shows that if you make the habitat available, in this case over geologic time, populations of ciscoes expand rapidly in response to that, but that the predators also respond. So as ciscoes diversify, lake trout similarly expand in populations. So it's really exciting, I think, from a fisheries restoration standpoint. If we restore the carrageenans, this provides some evidence that perhaps this will contribute to lake trout populations as well. Okay, so I talked about, I'm out of time, sorry. Patterns of variation, we see some concordance across data sets. Some of the important, excuse me, factors we think are light and depth. So there's evolving across this depth gradient. I didn't talk about depth, diet, and some of the others, but there's some other factors we think are important. Ole Seehausen's group talked about this idea of upward adaptive radiation cascades. These ciscoes, in this case, ciscoes and lake trout evolving sort of in concert, radiating in concert with one another. So as ciscoes diversify, it also contributes to diversity in lake trout. And we think this started about 65,000 years ago in the precursor to the Great Lakes. I'll just jump to the acknowledgement because I'm out of time. So there's so many people to thank. Many people have been involved in this research. I don't have them all listed here. I ran out of space in part. I want to say thank you in particular to Tom Dowling, who got me thinking about ciscoes several years ago, and also say thanks to the Great Lakes Fishery Commission, USGS, and Great Lakes Restoration Initiative for funding. And with that, if there's time, I'm happy to continue the discussion. Oh, and I'll leave you with this GIF of the receding glaciers and thinking about climate change over time.
Very nicely done, Trevor. No need to apologize for talking about the broader ecosystem or the food web in terms of lake trout. Really cool how they both lined up there at about the same time and how they responded to that increasing habitat. Fantastic story. So hopefully we have plenty of time for questions. You can do this a couple of ways. You can just pop your camera on. Hopefully I'll see you. You could also post a message in the chat. I'll try to make sure I see that as well. And the goal is to kind of continue our tradition of having I mean, there's no silly question, especially when we're talking about genetics, which is a complex topic. We have someone very smart here to sort of walk us through it. So, well, you're going to get a first one from Amanda. She didn't even give me a chance to ask a question, so it must be burning. Go, Amanda.
Well, I mean, Bo, feel free to ask your question if you would like to, because I could probably just sit and spend several minutes chatting with Trevor about all the stuff that you presented. But I just think it's really fascinating how different the lake whitefish versus the cisco genome was, and how much recombination there appears to have occurred across chromosomes. And so I'm curious how important you think, as you know, we're collaborating on a lot of this work and both moving towards looking at the genome level adaptation in this species complex, how important you think it is to start getting genomes for the other species to see whether or not, because everything is currently getting mapped to the Artidae genome, could we be missing important things, or important areas of relatively large chromosomal recombination between Chi or, you know, Chi and Artidae or Hoi or one of the other extant species?
Yeah, that's a great question. Thanks, Amanda. We were surprised too. I had no idea lake whitefish would be that different from cisco. I'm hoping it's not, because we have some errors in the assembly, but I don't think it wouldn't look quite like this if that were the case. So that's about 9 million years divergence time, if you believe those numbers, between these two species. So I think that's going to be much worse than the ciscos in the narrow sense, in terms of genome level differences between those, because the time scale is much finer. If you believe our number, it's 65,000 years or so. Even if it's 1 million years, it's much less divergence time. So hopefully those are not so different. But there are some chromosome level differences if you go back to these karyotype papers. There's actually polymorphism. What I didn't show in that Phillips et al. Paper, they find in northern, I think it was, I'm not sure which population, but they find polymorphism in one of the Artidae populations, Huron or Superior. Some of the individuals were heterozygous for this large chromosome level difference. And that was actually because of recombination between some of the chromosomes. So I think we're going to find the sample size in those studies was really small because it was difficult work. But if we go through some high throughput approaches, we might be able to see some of that. Ultimately, having genomes of these I think would be really helpful to give a shorter answer to your question. Yeah.
Okay. I see Owen's hand up, but let me just briefly jump in, Trevor. So I liked how you sort of walked us through how you're doing this detective work, how you are those frequency plots, and you were looking at the areas of the chromosome that had really high FST values, and then seeing what that gene was, I think is how you explained it. Do I have that right?
Yes. Yeah.
Okay. So can you do it the opposite way? Can you think about, and really cool that you found things like eye development or lipid, but could you also be thinking about things that, I don't know, like, for example, gill rakers. Gill rakers are something we know that's really different, or reproductive timing. I mean, we've got to have some sort of reproductive isolation, and sort of the descriptions of timing of spawning seem to differ, right, across especially the deep water ciscos. And I think for some salmonids, they found genes associated with run timing, for example. So can you kind of flip it the opposite way and say, okay, these are genes in other salmon that have been important, and go look for that in these, or does it not work that way? If that question makes sense at all.
Yeah, I think it's a great question. We sort of, I mean, we actually went back to an old talk, and we had rhodopsin was one of our like example candidate genes that we thought might differ. So, but more often than not, we're wrong when we pick the gene ahead of time. In part because if you look at some of these physiological pathways, there's many different places where like selection could occur. It might not be the gene itself, but it might be the receptor of that gene, or it might be some other part of that process. But also because these are ancient polyploid fish, there's also many different copies of different genes. So they have some of these gene families are large. So which one is the causal one is difficult to note. But I'll say that I see Rishi has his hand up, and their paper was that last month showed one of the genes that you'd absolutely expect for gill differentiation. We don't see it. I did look for that gene in these, but we haven't seen it yet. But I like the approach, of doing it the other way where possible. We'd like to do it mechanistically as well, to look at developmental series and look at how they're actually expressed and do the full proper functional experiment. But
Perfect. Okay, thank you.
Good question.
Well, let's get back to order then. Owen.
Yeah, I just had a sort of comment questions. One was the analysis showing the change in population, effective population size is pretty interesting, especially with the overlay of the lake trout. There's many parallels with what you have seen, perhaps with the rhodopsin gene in ciscoette. I had a graduate student with Tom Hrobick, and we looked at ciscoettes and did electrocardiographs of the retinas and so on, and determined a couple things that again, like Kai, they are blue shifted, and also they are lacking in cones, so they're pretty much monochromatic vision, highly adapted. And we found very conservatively, if we compare this with humans, at least 10,000 times more light sensitive than the human eye to low light level. And you might have also remember that the extinction and light shift in color occurs at depths between 80 and 100 meters, which is sort of the cutoff zone between leans and ciscoettes. And leans can see color, they have cones, and ciscoettes do not. So I find it kind of fascinating. You see these similar evolution of this capability. The other thing too I see in that, when the glacial maxima, the sky eye really took a big hit, losing a lot of that deep water at the glacial maxima. So their deep water habitat became far more limited, and that's kind of fascinating as well. So that's all pretty exciting stuff. And the other thing I would just point out is, in the work I've been doing with Amanda with our morphometrics and genetics, we see that same issue that Xenithicus, at least in Lake Superior, seems to be not well defined morphologically or genetically. It's kind of very messy. So it's good to see that we're seeing similar messiness, so to speak, between these groups. So, so I'm very, very excited about your work, Trevor. Thanks so much.
Thanks for your comment, Owen. That's really fascinating stuff. I'd love to see those the lake trout papers, and so hopefully we can chat more about that. That's great.
Okay, thanks, Rishi.
Hey, hi everybody. Thanks, Trevor. It was a really nice talk and a perfect kind of primer for me for cisco population genetics. It's really amazing. I guess I could ask you questions kind of all day, but one question, since we're on this slide as well, is when you were looking at the genome, what was your feeling about the kind of duplication level? Or, I mean, one thing that immediately came to mind when I saw this plot was, do you think this is the result of variation in the rediploidization of the genome compared with lake whitefish? Or, I mean, having done a genome assembly myself, I've become a bit skeptical about how challenging it can be for these very repeat-rich genomes. So I'm just curious what your experience was with that. And then maybe if you could mention a bit how you think moving forward we can kind of deal with this problem of highly duplicated genomes and resequencing data, which is something that I've been thinking about a little bit recently as well. Thanks.
Yeah, great question. So you're you know more about this than I think anybody with your experience with the Balchin genome, if I have the name right. They're humbling. It was humbling to work on this genome assembly for both myself and Nate, who did all the work on it, and this is still ongoing. It's a really difficult task because of this genome duplication, and we're finding, I think, in large part similar results to what you found. Like, there are these tetrasomic regions that are really difficult to pull apart. Some of which might be real biologically. It's not just a computational problem, but it's that the chromosomes are actually currently sharing information in some cases. We had the great benefit in our case, I can't remember if you had this benefit as well, but we had the linkage map that Danny Bloomstein and Amanda Akis, and I think Amanda was on it, I'm not sure. Wes Larson's group worked on. So that linkage map has been really helpful. Louis Bernachet's group and Eric Normando help also integrate that into the assembly. So that helps ground truth some of it as well. But it's a challenge for sure.
Yeah, in that case, maybe it's a bit better. I think we tried to use our linkage map that we did have, and it caused me more headaches. So we ended up kind of abandoning it a little bit. But that's great to hear that you had some success with that.
It's tricky when the two conflict. You have the high C data and the linkage map, and so how do you reconcile which one's right? So it's difficult.
Right. Thanks for that good question, Rishi. Mark Ridgway.
Hi, Trevor. Thanks so much. That was just fantastic stuff. There are so many questions, but I'm going to go to the 65,000 years ago question. That was nowhere near the glacial maximum in terms of timing, as far as I understand the glacial story around the Laurentian Great Lakes, and it continued through the maximum. What's the crazy speculation on that? Is that when populations really became isolated from each other, whatever that habitat isolation would have been, a refugia question? And they seem to not get squeezed by expanding glacial, by glacial expansion and perhaps loss of refuge or declining refuge, as the effective population size might imply. They just kind of sustained it through it. So I'm just interested in just like crazy speculation. What might account for that?
Yeah, and I think speculation is a great word for that. Thanks for that question, Mark. This was surprising to me too. I thought the divergence time would correspond to at the last glacial maximum. Then you would start to see this diversification. So this idea that that divergence predated the last glacial maximum was a bit surprising. I didn't know my history of the Great Lakes, and I'm still learning about this, the geologic history. But this time period, this 65,000 years time period, there was a lot going on. It's between marine isotope stage 4 and 45,000 years. They switch between the terminology here. But you can see these are the isotope levels, which gives some indication about the temperature at the time in marine areas, but basically how cold was it on the planet at that time. And you can see these lines are shifting over relatively short time periods, several millennia even. The environment differs a lot. The actual Great Lakes Basin, parts of it are really old. So Lake Superior sits on the rift that's a billion or so years old. And I reached out to a geologist, a colleague who works on this, and she explained to me that the actual carved out parts of the basin, it's not all just from the last 10,000 or so years, but the actual parts of the basin, depending on the lake, have had previous periods of water in them. Somebody can please chime in if I have this wrong. But the 65,000 years ago time, if things were warmer, there may have been water there and some time for more Great Lakes-like kind of conditions. Then they go down into the glacial refugia, populations crash, and then the expansion subsequently. That is wild speculation, but those basins, I guess, have been around for longer than that.
Yeah, no,
occasionally with water in them.
Thanks so much. I mean, yeah, it's wonderful to actually speculate what might be going on. Thanks so much.
Okay, good question. There is a question from Sarah Salisbury in the chat. It's got several parts to it. Do you have a sense, Trevor, why the transcriptome and whole genome show different things in terms of the degree of overlap between Xenithicus and other species? Overlap with all species using whole genome, but mostly, I believe, with Hoya in the transcriptome. Were they different samples or temporarily differentiated? Maybe just stop there for that part of it. And then she's got a second part that has to do more with the introgression question. But
yeah, that's a great question. I think to some extent, the way that these analyses are conducted based on sample size differs. So the statistical power to some extent differs. That shouldn't affect the PCA, but and then the identification part of it is more difficult. With transcriptomes, we were being very careful because they were expensive at the time, and you had to get it right with that smaller number of samples, and we still didn't really get it right. But some of those might be just misidentifications across. It's challenging to do this work because to get the transcriptome, you have to do the dissections right away. So you have to do a quick identification, do the dissections, and then hopefully you got it right. But to have high quality RNA, it's difficult. I'm not sure if that answered the question.
And then her second part, maybe she could jump on if she has additional ones, but has there been any evidence for selection against adaptive introgression with Xenithicus? And I think you showed it mostly with, oh yeah, so you showed it mostly with Hoyi, but maybe why didn't it happen with the other species, with Arteti or with Kai? So any thoughts on that?
That's a great question. We haven't tried to dig too much into the adaptive introgression just in general, but the adaptive piece as well. We're stuck between trying to decide to what extent we're just misidentifying things, and so it's not really true introgression. It's just we just don't have the names applied correctly. Between that and actual introgression, and then the third possibility is that there's also, because this is still a relatively young system, that it's incomplete lineage sorting, and so maybe you just have some of these old variants. And actually now I just thought of a fourth one, which is this dual glacial refugia. So some of the genetic variants are coming from Mississippi and some are from Atlantic refugia. So yeah, it's really complicated to pull those apart. I think once we have a better handle on that, Amanda's and others are working on developing better markers for the species, along with the morphology with Owen and others. If we can get those down, then we can start trying to tease apart the adaptive introgression piece in more detail.
Yeah, she says, yes, thanks. I was just wondering if maybe the reason there was less overlap in the transcriptome between Xenithicus and Arteti and Kai than across the whole genome was due to selection against introgression in the protein coding regions.
I see. Okay, I missed that part of it. Yeah, that's really interesting. We haven't put the transcriptome data back onto the genome to see. So we were doing it de novo at the time, but now we could map to the genome and see if there's any kind of patterns there. I like that idea. Thank you.
Yeah, and she says it's really interesting about the multiple refugia. So.
Sorry, I had one thing I forgot to mention relative to Mark's question. Sorry to interrupt. Bernachet's group, Laporte et al., had this Science Advances paper recently on Lake Whitefish, trying to figure out the timing. And I just wanted to add that they also came up with this 60,000 year. There's some references for that as well, but there seems to be some kind of concordance across different taxa. I don't know if that's coincidence or not, but another piece of that puzzle.
Yeah, no, that is compelling. Multiple lines of evidence. Nikki Berry. Hi, Nikki.
Hey, hi, Trevor. Good talk. This is really interesting. I was wondering for the short wavelength options that you talked about, it's kind of interesting to me that those were like kind of the second highest like peak in that grouping of peaks. So I think those are the ones, if I remember correctly, I think those are the ones that are actually used or thought to be used to detect like UV wavelengths and UVB area. So I was kind of wondering if you think that those opsins might be also kind of changing in the similar way as which the rhodopsin opsins have been changing. And if so, like if you think, if I understand right, you're thinking that some of these changes are also consistent with the changes in the depth or the light environment in which the fish are occupying. So I wonder if you could look at the UV environment too, especially like comparing like Superior, like Ontario Erie, right? And seeing if that also would be changing, or if the fish themselves might actually be a little bit too deep. I don't know if you have anything to say. Or
yeah, I was really excited to see your work on UV because it in light of these data, yeah, it'll be really fascinating to see. In the case of shortwave sensitive opsin, they were not as strongly differentiated across species. That's why I didn't talk about them, is because there's more noise there relative to species, but they could still be important, for example, developmentally. What we haven't looked at, for example, how expression of these opsins have changed throughout development. So in these early life stages, if they're in shallower water, if, for example, maybe they're expressing these genes. So maybe it's not at the sequence level, but the actual expression and what that gene is doing across different life stages might be useful. But yeah, I look forward to hearing more about the UV story and thinking about how this all might relate.
Dr. Ackes.
I just had a follow-up question based on Nikki's comment that I remembered, which is, I can't remember if you said anything, but do you happen to know in the genome that we have where the opsin gene would fall that you saw fixed in Chi? And have you taken a look on that chromosome to see if that's related to any like high levels of differentiation?
So that's a great question, and I should have been prepared for that. I don't remember where it is. We've looked at it before, but it was in an earlier iteration of the genome. So things, as we redo the genome assembly, things have moved around. It didn't come out in any of the like peak FST peak sort of analyses. We didn't just like happen to see it, but we haven't really carefully looked for it. That's a great question, though.
I guess another question related to it is, I'm assuming just for sanity and computing sake, you've thinned the SNPs that you showed in the Manhattan plots. And have you had a chance yet to, or you or Nate had a chance yet to maybe just reduce to that particular area of a chromosome and maybe back off some of the SNP thinning to see if it's actually an inversion that you're dealing with, or if it is very much a like gene specific selection, anything like that yet?
Yeah, great questions. Nate's been working on, I'm not sure if Nate's on the call or not, but Nate's been working on the thinning, SNP thinning part, which turns out to be really computationally intensive. There's 4 million SNPs, so winnowing that down, it takes a lot. So based on linkage disequilibrium. I think he just got results for that. Nate, feel free to chime in if you're on.
He's on.
Hi, Amanda. Those FST plots are actually with all SNPs.
Are they really?
Yes. So we're working on the low coverage whole genome sequencing and the moderate coverage one that was used for the FST plots there to do some LD pruning. But those in particular are all SNPs that we found,
Okay.
all 4 million.
Okay, that's really good to know.
So maybe while we see if we have any further questions, it really is a puzzle that you posed, Trevor, about the potential misidentification, or just to put it another way, the similarity in how some of these deep water ciscoes appear. And so how do we, I guess one solution might be to just take samples and see if they sort, and then see if there are morphological characters that associate with them, where you sort of have no a priori assignments. But that still doesn't help us in terms of a management perspective when we don't always have the quick ability to use a sort of a genetic test. So I don't know. I mean, being mostly a Lake Michigan biologist, in my previous years, we didn't really have to deal with this, but we do have several Lake Superior biologists on the call, and I just wonder how people are reacting to this sort of lack of concordance or the lack of separation with Xenithicus more broadly, because I think we're seeing this in Amanda and Owen and Amanda's data set. We're seeing it in your data set, and it's just it's a really unique challenge, and I don't know really how we go about trying to figure out how to separate out the misidentification from just the pure or more blind more fair assignment somehow. No, just wondered if that sparks any reactions or questions from you or anybody else that has experience in the field with these fishes.
Yeah, I'd like to defer to Amanda, but just to say that her GT-Seek panel, I think, is going to be really useful for this exact purpose of, because it's designed specifically with careful, large sample sizes and carefully with respect to this. So I'm curious what Amanda has to say.
Yeah, it's a real challenge, and you know, you've talked about it, Bo's talked about it, and Owen briefly mentioned it too. And I will just say the original RAD data set where we had a couple of Xenithicus samples, I think, I might have this reversed, but I think like 3 of them, out of the 4 we got to genotype with RAD, 3 of them fell out as bloater and 1 of them fell out as Kai. Nothing was unique. And that's a pattern that's been repeated with this whole genome resequencing data, with the GT-Seek data that we generated for Owen's project, which included almost 2,000 fishes from Lake Superior in the cisco species complex. And so, but with Xenithicus, there was nothing strongly distinct in it. And so I think Trevor did a nice job of sort of discussing the possible reasons why we see that with Xenithicus. I will say I was on the boat and they have a key for identifying Xenithicus. And for somebody who doesn't have experience, or possibly even people that do have experience, the differences are very, like between bloater and Xenithicus, for example, it's like, well, the pectoral fin length is like 65% of a certain ratio, and it's, or, you know, 68% on average in bloater. Like the differences are really, it's difficult and it's a challenge. And so what we're doing to try and understand this better, to try and address some of these hypotheses, and I will just give a plug for Moises' paper that just came out in Molecular Ecology, where they discuss some of the possibilities in their discussion section about what's going on with these Xenithicus. And it could be that there was some integration. And so what we're seeing is some integrest thing that looks morphologically what somebody thinks might be Xenithicus. And so one of the ways Owen and I are trying to address this is to track down historic samples, historic scale samples of Xenithicus, to see if this pattern of like, sometimes it looks like bloater and sometimes genotypically it looks like bloater, sometimes genotypically it comes out as Kai, to see whether or not we see that same pattern, or if there's any kind of discrete genetic clustering in historic Xenithicus, to try and tease out what that issue is. Because it's possible that it's an arbitrary, you know, it's something that's an environmental phenotype, environmentally driven phenotype, where there might be multiple species that can, along the distribution of morphology in those species, converge on this in a certain environment, and then it's being called a different species when really it's not. Or if it is truly something discrete, or historically was truly something discrete. So it's a very interesting question, and we're trying to address it to figure out what's going on with the Xenithicus problem.
And maybe you can just add, Amanda, when you look at the historical Lake Michigan Xenithicus,
yeah, the important point. So with historic scales from Lake Michigan of extinct or extirpated species, Xenithicus from Michigan, historic Michigan samples very discretely cluster in their own thing. So we had some data from Lake Superior where we were looking at some clusters, and we found that there's right now putative Reichardii in Lake Superior that have been morphologically ID'd as Reichardii, and they cluster distinctly. And we put in the historic Michigan data, and it falls right on top of it for Reichardii. So we thought, okay, let's put Xenithicus in to see if it's like if that shows up with anything that we see in this large data set with this GT-Seek panel. And the Lake Michigan cluster nice and discreetly in a shared data set, but it doesn't seem to associate with anything that we're finding and calling Xenithicus and Lake Superior. And I will point out too that in your whole genome data, Trevor, it was really nice to see your Huron cisco falling right on top of, at least on PC1 and 2, falling right on top of the Lake Superior cisco. So species with species within the Great Lakes. And then this sort of phylogeographic split and differentiation between what's in Lake Nipigon versus what we're finding in at least the upper Great Lakes of, Michigan, Huron, and Superior.
Okay, thank you, Amanda. Rishi has his hand up again.
Yeah, sorry, you'll have to tell me to shut up if everybody wants to leave. I was just going to ask what the genome-wide FST is actually between these species. So, like when we're on the topic of what's a real species and avoiding that whole hellhole of a discussion. But yeah, I was just curious from your estimations.
Yeah, I'll take the first part of that question and maybe ignore the second part.
I think that's probably better.
Genome-wide FST is something like 0.001 or something. I may not have the exact number. It's incredibly low. But nonetheless, you see these regions of the genome that seem to be important.
Yeah, thanks. I think it's an interesting discussion that you're having because, I mean, we have the same sort of taxonomics. Yeah, the risk of a circular argument also with the alpine whitefish. And, I think this is a general problem in population genetics, actually, that many of us have a subconscious tendency to select the most phenotypically distinct individuals even, and then to try to draw conclusions about populations when actually it's not very convenient to sample intermediates, whether that be genetic or morphological. So it's great to hear this approach. Thanks.
Thanks for that perspective, Rishi. I'm gonna have to go and read your Nature Communications paper. So. All right. Is everybody feeling talked out? Your voice held up, Trevor. Trevor joined and his voice was a little bit raspy, but it held strong the whole time.
Thanks, Bo. Thanks, everybody, for coming.
Yeah, great talk, great discussion, and hopefully we'll have another one next month with, I guess Rishi could say her name better.
With Feline. Yeah, you're all in for a treat. I'll try and join as well. It'll be a great talk.
All right. Very good. All right. Thanks, everybody. Have a great weekend.